Hello, my name is Indresh Srivastava, and I am going to be giving this masterclass on the vaccine characterization. So, um, yeah. So here is my slides. I think for you to take a look at that. So the title slide, I'm the vice president at Novavax. I recently joined Novavax. The second slide, I just want to show that there is a continuing race between the scientists and the pathogens. I think we scientists, we try to make the vaccine so to control the disease, and the pathogens, I think they try um, to infect us so that they can cause the pandemic. In case of several times, I think such as Spanish flu of 1914, malaria, cholera, polio, and more recently, I think for COVID-19, I think the pathogen won and they caused the pandemic. Then I think as scientists, we came back and we developed the vaccine or the effective drug. So it is very important to understand that there is always a race for survival by the pathogens, and there is an effort by scientists to control the disease. This is how the presentation is organized. So we would like to talk to you or share with you my vision and challenges in developing the vaccine and the vaccine platforms that are being used for developing the historical and the modern vaccines. Then the rationale for, or what are the reasons for performing vaccine characterization, what type of assays can be used, and then how these assays can be applied for the bioanalytical compatibility, which is critical when we are doing the vaccine production at multiple sites, when we are licensing the facilities, new facilities, or when we are developing the new processes. I think we obviously don't want to do the clinical trial, or we should try to avoid doing the clinical trial in order to manage the cost and the timeline. So again, I think these are the platforms that have been used for the historical vaccine. They are simple. It's a killed vaccine, so live attenuated vaccine, isolate, inactivate, and inject. This is another approach. I think these are all based on the Pasteur's principle for vaccine development. I think at probably mid to late 20th century, I think people started using recombinant vaccines. Again, this is the slide that shares the modern vaccine platforms. I think one is the DNA delivery, vector-based, recombinant protein, and mRNA. It's a viral replication particle, VRP, and then virus-like particle, that is VLP. I think we all are fully aware of the accomplishment of mRNA platform that has developed the COVID-19 vaccine in probably the record time. Also, the vector has been used for developing the COVID-19 vaccine, I think both by Johnson & Johnson and AstraZeneca. And then the recombinant vaccine is not too far behind. There are two companies which are playing a critical role in developing the recombinant vaccine for COVID. One is Novavax, which has completed phase 3 trial, and then Sanofi Pasteur has initiated the phase 3 trial. So there's a lot of work has been done rather very quickly on these three platforms. Effort is also being undergoing in terms of looking at the DNA, looking at the combination approach. The pluses that we are seeing for each platform, this is the perceived safety and potency for individual platform. This is my view. It does not represent either Novavax or any other company's version. This slide shows that understanding the correlator protection is very, very important for successful development of the vaccine. When we understood that antibody is a correlator protection, we have developed very quickly a number of vaccines. It has been a little bit of challenging to develop the vaccines where the correlator protection is cell-mediated or T cells. But in recent years, I think Shingris has came up where the correlator protection is totally T cell or a polyspecific T cell and epitope. And I think this is what is probably a good example. It may be because of the adjuvant that the vaccine is using, but nevertheless, I think it's a very good development for the vaccine development. Other part which is very important for us to understand it, that developing a vaccine is very successful when there is little or no antigenic diversity or variability, as it is shown by this red panel here. But once the variability starts, I think developing the vaccine is a little bit more complicated. In terms of influenza and COVID-19, they are together in terms of antigenic variability. And as I said on the left-hand side, that they have comparable antigenic diversity for COVID. New variants are coming up. And these variants are not hugely different, but they have the mutations in the critical receptor binding site. My perception is that the COVID vaccine in the long term will be a seasonal vaccine similar to the influenza vaccine. Moving into the part that why it is important to perform vaccine characterization, there are a number of reasons, but the most important reason is that we need to establish batch-to-batch consistency, means the quality of the vaccine is maintained whether the vaccine is produced at one site or multiple sites. All the batches has a similar quality attribute. It is important for the vaccine efficacy and the safety. It is also needed to demonstrate the stability, formulation development, develop the potency assays that can use to reflect the efficacy of the vaccine. The structural characterization is required for monitoring the conformation of homogeneity. And since viral proteins are glycosylated, therefore glycosylation profiling is critical. We don't want the glycosylation profile change either during the upstream or downstream that may impact the potency of the vaccine. Monitoring the exposure of the critical functional epitope is also necessary. And then last but not least, I think we need to demonstrate that the assay that we are using, they are stability indicating, and then how to use these assays for bioanalytical compatibility. Then in this section, I think I focus on in terms of analytical tools that can be used for vaccine characterization. As I'm listing here is that the different steps, at different steps, we need different set of assays, and I will walk you through. For vaccine characterization, both characterizing the product and impurity is very, very critical. Characterizing the product is critical to maintain the vaccine efficacy, and characterizing the impurity is critical for vaccine safety. For characterizing the product, I think we do perform in-process testing. And in the in-process testing, we focus normally on doing the STS page or determining the purity. We also look at the degradation profile and how the product is becoming more pure with the later stage processes. So we look at the homogeneity, and also we look at the potency by doing an ELISA. And then comes the second main part, that is the characterization of vaccines. So essentially, first we do the immunological characterization that is going to be based on ELISA, Western blot. Then we do the purity by STS page, size exclusion, the reverse phase, and mass spec. And then we start using the biophysical methods for characterization of the vaccine, such as CD spectroscopy, light scattering, differential scanning, calorimetry, and then reverse phase, tryptophan fluorescence, glycosylation, and then transmission electron microscopy. Again, I think not all these assays need to be performed, but this is what we need to, or we need to decide early on in the project that what assays are critical and what needs to be performed. In terms of the product release, I think we need to focus on potency, identity, purity, and then the residuals in terms of host cell DNA, in terms of the host cell protein, in terms of the bioburden, in terms of endotoxin, and the process residuals. So that combines the aspects of vaccine characterization and release testing. Again, I think as I mentioned it, that change in the tertiary structure, secondary structure, aggregation state, degradation, all these things will compromise the vaccine potency and develop the suboptimal vaccine. So therefore, I think monitoring all these aspects by different characterization is critical for successful vaccine production or manufacturing. I will give you two examples. One is based on chikungunya vaccine. Another is based on the trimeric HIV protein. This is the work that we did at Novartis, and this is the work that was done at VRC when I was at the VRC. So VLP has three proteins, E1, E2, and capsid protein. Together, there are 240 copies of each protein on the VLP. And the mass is a giant, it's a big protein. It's about 67, 68 nanometer structure. VLP, the trimer is the three copies of HIV envelope protein, GP120. They come together and then produce the trimer. The mass is about 400 to 420 kilodalton. The first part we want to show is the purity and identity. And this is the example is chikungunya vaccine. The first thing what we did is a STS page analysis. You can see that here we have E1, E2 protein. This is the capsid protein. All three proteins are present. And then we showed the Western blot analysis to demonstrate the identity of the vaccine. This is the example that I'm showing you for HIV, the trimeric protein. This is the native STS page, native gel, where we are showing this is the trimer, this is the monomer. The ratio is about 60-70% trimer and about 20-30% we are getting about the monomer. I think these trimers, they do dissociate during the purification and formulation process. The same data is here. This is what the trimer looks like. This is what the monomer looks like. And this is the reverse phase assay. Again, I'm showing you the data on 2 batches. Next, we want to show you about the potency, how we develop the potency assay. Potency assay is rather main aspect, but it does take time to develop it. So in this case, what we did, we developed a number of monoclonal antibodies. We tested them for their neutralizing potential, and then we used them to develop a capture ELISA. And here are the 2 monoclonals that are being used for in the potency method. And as you can say, that these are the 4 or 5 batches that we have tested and characterized in terms of the potency. This is what we have done, the BCA on those drug substance batches. And you can see the ratio between ELISA to BCA is between 80 to 120%, which is very, very good, I think, in terms of the products are consistent. Then we show the homogeneity. I think so we have tested all these drug substance batches in the size exclusion HPLC analysis. And you can say that all these batches can be really overlaid. The peaks are symmetrical and there is not change in the retention time. We calibrated the column using these 2 standards here. Next, we want to show you that the DLS analysis that we have done. DLS is dynamic light scattering. It is being used to develop the particle size. And here we have overlaid 4 batches of the VLPs. And you can see that there is a perfect overlay. The sizes are between 66 to 68 nanometer. So it is very small variability, and the polydiversity index is extremely low, suggesting the high quality DLS data. Again, this example is for trimeric protein. Here we are using multi-angle light scattering in conjunction with the size exclusion and the refractive index. And what we are trying to show you here is that there are two main peaks or components. One has about 420 nanometer, and the other is slightly smaller, which is 3.56. So I think this may be due to maybe slight aggregation is coming out here that may be increasing the size as calculated by the software. Nevertheless, I think these are the two batches, and both the batches are giving us comparable data set. For VLP vaccine, because it has 3 proteins together, it has the E1, E2, and capsid protein. We wanted to make sure that we are able to quantify those. So we have separated out. We treated the VLP and did the reverse phase HPLC analysis. We can see that all 3 proteins are there, E1, E2, and capsid. And this is what we have done to demonstrate to identify them using the Western blot. Again, I think we wanted to do the transmission electron analysis for the VLP. And as you can see, with the VLPs are very homogeneous. We did the carbohydrate analysis, and it is shown here is that predominant carbohydrate that we are detecting on these VLP is the terminal mannoses. For HIV envelope protein, we are seeing both the terminal mannoses as well as the complex carbohydrate. So this is a differential glycosylation profile. But the good news is that the profile is consistent for both these 2 batches. Moving on to the next set of tools, and this is for characterization of the secondary and tertiary structure. So I will just walk you through. So the first method that I'm going to show you is the CD spectroscopy. And as you can see on the left-hand side, I think that vaccine or any protein, depending upon what it has, it may have different profile. Look at the black profile. This is where if you have predominantly alpha helix, then this is what the profile you may see. If you have the beta sheet, that you will see a profile similar to the red line. And if you have random coil, then you will see a profile similar to the green line. So these are the 3 distinct profiles you get if you have pure alpha helix, beta sheet, and random coil. This is an example. If we take the chikungunya VLP, I think it seems like it is a combination of alpha helix and beta sheet. But if you denature it, if you heat treat it, then you lose the secondary structure. This is the example that I'm showing you for the flu vaccine. And this is the work that we did at, the team has done at Novartis. Here are the 2 aspects. One, the protein is, the vaccine is kept at 4 degrees. And here the protein is kept at 50 degrees. 4 weeks, 4 degrees, it maintained its secondary structure, but 4 weeks at 50 degrees, it lost its secondary structure. So it is a stability indicating. It does, it can be used for characterizing the vaccine, the protein, and also for stability determination. Here is the example for chikungunya vaccine. In this vaccine, it was treated from 25 degrees to 60 degrees. And here, I think you will see that we are losing the elasticity with the increase in the heating temperature. By the time we reach about 55, the structure is gone, secondary structure is gone. So once again, I think it could be used for the post-degradation study. It can be also used for the real-time study, can also be used for the accelerated study. The method has a high potential. Again, showing you the tryptophan fluorescence spectroscopy. Here, I think on the left-hand side is the conceptual idea. The tryptophan, the intrinsic fluorescence, I think the intrinsic fluorescence state is red. So they get a maximum spectra probably around 340. But as the protein gets oxidized, the tryptophan changes the color to greenish yellow. And that's where the spectra is going to be shifting to 350, 360. Here is an example. The blue line, this is the vaccine, is the time 0. And then it is 4 weeks and 6 weeks at 50 degree storage. and you can see that the spectral profile has started changing from 357, 358, like here, or 360. Then it is really, really going more to the right-hand side. And here, I think we are getting two transitions. One is about the same, probably around 360, 370. Another is about 420. So again, I think this is a stability indicating it can be used for the force degradation, and the assay is a stability indicating. It can also be used in conjunction with either ELISA or octet to determine the potency. So this assay has a lot of value. Next part, I want to show you, give you an example on the microcalorimetry and what it can be used for. So there are two methods. One is the isothermal titration calorimetry. This is called ITC. And this is a differential scanning calorimetry. This is called DSC. So isothermal titration calorimetry is being used if you want to look at the receptor ligand interaction. And for example, in HIV envelope, we have three CD4, putative CD4 binding site. In case of COVID, we may also have three ACE2 receptor binding site, and that can be used to demonstrate if all the three sites are open or accessible or not accessible. Differential scanning calorimetry, I think it does provide us an idea about the thermal stability or unfolding of the protein. Here are the example, I think in the left-hand panel, we are showing you here that in case of HIV envelope protein, which is a trimer, where we have all the three binding sites for the CD4 are open in the trimer. Again, I think if you create a different structure, the binding to CD4 is changing. And I think if you look at it, this is what is the monomer. This is what the trimer looks like. And these are the two versions where the V2 loop is deleted. So all these binding profiles are different. We published this work, so if you're interested, let me know and I'll send you a PDF paper. This is where the DSC, in the DSC for the HIV trimer protein, we saw two transitions. We have taken the protein to the HPLC analysis, and we are seeing two different profiles. One, this represents the aggregated state, which has the higher thermal stability or TM, and one is the main area where the protein is in correct conformation, and that's where we have the main transition. So again, I think this method can be used for determining the stability, determining the homogeneity, and how to characterize the thermal stability of the protein. Another way to study the thermal stability is by doing the differential scanning calorimetry. What we have done, we developed this 96-well format where the protein is the different concentration of the protein is put it together. It mixed with the fluorescent dye, and then it is being heated in the fast real-time PCR machine. As the protein is being unfolded, the fluorescence dye, it does interact with the hydrophobic patches and then give rise to the fluorescence. We have used this technique to determine for the force degradation study. The first example is if we boil heat treatment. If we boil the protein, we lose the structure as it is shown here. If we treat the protein at low pH, we destabilize the structure. It can be degraded. But if we do the low pH treatment, then neutralized, and then test it for differential scanning calorimetry, we can get some structure back. If we do the oxidization, we increase with increasing oxidizing agent, we saw the denaturation in the protein. So the technique can be used very, very effectively in the stability indicating assays in terms of measuring the force degradation. And also, the technique can be used for the potency. So in summary, I think we are using a number of techniques, both immunological, that is the Beer code and ELISA, and then we are also using the biophysical method that is ITC, DSC, and CDS spectroscopy. We are doing the triple analysis to determine the purity, And we are also looking at the glycosylation profiling. And in addition, we are doing the structural characterization by negative staining. So in terms of, this is a slide I just wanted to include you for you to understand that when you are developing the assays, I think you need to keep in mind that we don't need to use all the assays for everything. We need to determine that which assay is going to be critical for which aspect. And I think this is what we have listed the assays. We have listed the type of vaccine, the viral vaccines, and this is for the bacterial vaccine. So hopefully that may be able to help you in terms of developing the assays for characterizing your vaccine. And then this is a slide that we are using it to demonstrate what is the compatibility, bioanalytical compatibility approach. So we use a combination of release tests that are being performed by QC. Then the characterization assay that are being performed by analytical development. We combine those and develop the compatibility, bioanalytical compatibility approach. This is a very comprehensive. Again, I don't need to really rehash the conclusions, but I have shown you why it is important to do the vaccine characterization, what the methods can be used for characterization, and what we and how to use the methods for developing the bioanalytical compatibility approach. Last but not least, I would like to acknowledge my team at VRC, at Novartis, and at Protein Sciences. And I would like to acknowledge the leadership which helped me in terms of allow me to accomplish the work that we have done. Thank you very much for attention. Please feel free to reach out to me if you have any questions. Thanks again.